Okay, so the title of my talk was initially Neuronal Imaging and Reconstruction, but as you see in your programs, it says, why does ICT need biology? So the short answer will be, well, to obtain data. So end of the talk, we are going to take some coffee. But I wanted to show you how the techniques we use to obtain this data, okay? So let's start by talking about our focus of interest, that is the brain, and more specifically the cerebral cortex. The cerebral cortex is the outer part of the brain, and it's directly involved in many aspects of mammalian behavior. And it's considered to be the part where most of our human behavior resides. Histologically, you see that the cortex is situated, as I said, in the outer part of the brain. It covers just a few millimeters depth, and it contains the main cells of the brain, the neurons and the glial cells, and the blood vessels. Macroscopically, you can see that the cortex is organized in layers. Depending on the part of the cortex you are studying, you have a variable number of layers, but the typical cortex is composed of 6 layers, being number 1, the outer layer, the closer to the surface, and the layer 6, the deeper one, in contact with the white matter. Maybe you all know all these things, but I thought it was a good idea to start reviewing this concept, because I noticed that most of you have a more informatics, computer background, so it's not a bad idea, just go back to what we are studying. So this is the organization. But what we know now about the brain is, in fact, a very recent knowledge, because it was not until the half of the 19th century where the nervous tissue was studied. Why? Because there were not appropriate staining methods to visualize the neurons. So they only see this kind of cells, and they did not know anything about the branches, about dendrites, or about axons. In fact, it was Kolika who first described this kind of cells in the nervous system. It was not until 1873 that Camillo Golgi, well, the slides can be seen very well in this projector. They were very, very beautiful, but I don't know if you can see them. So Golgi discovered this staining method that he called reazione nera, that was very new at his time, because it was the first time that he could visualize the complete neuron with all the processes coming out from a soma. And it was the first time that a neuron could be drawn. At that time, as you know, the biologists used the microscope, optical microscope, and just painted what they saw. And it's surprising how these paintings are so close to the reality, to what we see in the microscope and the images we take today with photographs, okay? What were the advantages of Golgi's reazione nera? Well, it stained only a small proportion of neurons, and that is a point that may seem a disadvantage, but in fact, this allows you to carefully study the morphology of each neuron. So you can distinguish all the features that characterize the neurons. And furthermore, it makes possible a classification of the neurons. But it was Cajal, Santiago Ramón y Cajal, who studied more accurately the nervous system and described it in his wonderful pictures that you have here an example that were very, very precise, and nowadays is still a reference for all the neurobiologists. He was able to describe the neurons, and even he proposed a directionality of the function or the proposed function of the neuron, as you could see in the image. Also, Cajal was responsible for the illustration of dendritic spines for the first time. He first described these structures in the cerebellum of the hen, and afterwards in many other species and cortical areas, both in human, in rabbit, in cat. And he described them as very thin protrusions of the dendrites. He, at the moment, didn't know the function, but he proposed to call them spines because they were very similar to the spines coming out of a stem of a rose. So dendritic spines were described for the first time by Cajal, and they were some kind of forgotten until the 60, 70 years of the past century when they were again rediscovered. About the classification, as I said, from the very beginning, neurons were classified into two big groups, the projection neurons that were pyramidal cells. They were excitatory, most of them, and they constitute about the 80% of the neurons of the cortex. They can be further classified based on the projection places where the axons go. So you have thalamocortical fibers, spinal cord fibers, etc., and for the layer where they are. Most of them, if not every of them, have spines on their dendrites. The other group are the interneurons. They are inhibitory. Their main transmitter is GABA, and they are only about the 20–30% of the neurons of the cortex. But this is a very heterogeneous group that contains many morphological types and many neurochemical characteristics. We are not talking about these neurons today, okay? So in summary, what we know about the neuron today has been obtained about the observation of images of neurons. So you have a cell body that contains the nucleus and controls the functioning of the whole cell. You have an axon that in pyramidal cells like this, in projection neurons, leaves the cortex and sends the information to another parts of the brain. There are also collaterals that make local connections with other cells in the cortex. Apart from this, and coming out in the other direction, you have the dendrites. The dendrites can be in the pyramidal cells classified into two groups, an apical dendrite that goes towards the surface, and a variable number of basal dendrites whose surface, both the apical and the basal dendrites, are covered by these dendritic spines. Why are important? Dendritic spines. Why are dendritic spines important? Well, because they are the main postsynaptic element of the neuron, and it has a correlate morphologically. I mean, if you take a picture of electron microscopy, you can see a dendrite here, the shaft of the dendrite, and coming out, the spine. Here you can distinguish the spine head and a neck that joins it to the dendritic shaft. And it's mainly on the spine heads where the synapses form. This is an axon, a terminal axon, which is making a contact, an excitatory contact, with the spine. As you can presume from the previous slides, the neurons are not only important by their morphology, but also by their function. I mean, these elements are prepared to transmit information. So, pyramidal cells form part of what is called cortical columns that are microcircuits that are arranged in columns coming from the layer 1 to layer 6 in the cortex, and whose connections are presently studied. We know that afferent thalamic fibers make contact into the central layers, especially layer 4, and also the surface layers. And these afferent thalamic fibers make the neurons in the column start a complex net of connections that we are currently studying, and the output is only one signal that comes out of the column, and it's the result of the interaction between external, intrinsic, and stored information. And this is one of the main goals, actually, to know how this connection and the composition of the column, the precise number of neurons that are present in these columns. So let's start with the techniques that we are currently using to imaging the cerebral cortex. So I wanted to start by reviewing the types of microscopy we have. You have optic microscopy, you have bright field to obtain, excuse me. You have images like, not like this, much better than this, but and the computer is wonderful. Where you have neurons, you need a staining that allows you visualize the neuron. Usually these are precipitates of some kind of salt on the structure you want to visualize because you are illuminating with white light, or you can use fluorescence. In fluorescence, you have a dye that is fluorescent, you excite it with a mercury lamp through a filter that excites the fluorophore, and you get the emission. You get this kind of pictures. Both bright field and conventional fluorescence give you 2D pictures that are maybe useful for some goals, but it's not what we use usually in the lab. We use confocal microscopy. Confocal microscopy is a very powerful tool in general in biology, but specifically for neuroanatomists. In this case, the source of light comes from a laser that illuminates the fluorophore, and you got then a PMT detector to recover the emission of the fluorophore coming through a pinhole. All this structure, what allows you is to obtain a single focal plane images. And this is very important because on one side you can get a series of images of the diverse focal planes that you have in your sample and have a 3D reconstruction, and besides you can use several fluorophores to stain different structures and see if they colocalize in the same focal plane, so if there's really a correlation between two stainings. And this is our confocal microscope, where we have obtained all the images that I'm going to show you in, well, no, not all, but most of them. If you want more resolution, no, this is a video explaining how this works. I mean, this is a neuron stained in green, and you have another staining in red, which corresponds to axonal terminals, inhibitory axonal terminals. So you have, what you see in this video is the stack of images running from the first to the last one, and you can see the spines in the dendrites, you can reconstruct it three-dimensionally, and you can see the colocalization of some of these dots, red dots, that corresponds to axonal terminals in close contact of the spines. Well, okay, if you need more resolution, you have to get another techniques that are in general called Superresolution microscopy, and that covers a lot of new techniques that I'm not going to discuss now. They go beyond the diffraction limit, and every commercial brand has his own super resolution technique, or you can go to electron microscopy. As you know, you have several approaches. You have transmission electron microscopy that gives you that kind of images that you can see here, but I have it in the computer. You can resolve very, very small structures like synapses here. You can see only dots and these stripes, but this can be studied very easily and recognize all the structures. You have scanning electron microscopy that gives you these typical images of surfaces at very small detail. These are blood vessels of the brain. And in the lab, we use this VSAM technology. The VSAM is this kind of microscopy, and it has been developed very recently. It has two beams. One is 5 milling beam that is focused ion milling that in fact mills your specimen, makes a hole in your specimen. You have another beam that is scanning electron obtains images from a scanning electron microscopy. So you have a gallium ion a gallium beam milling your specimen, an electron beam taking images. So at the end, what you have is a stack of images that allows you to reconstruct three-dimensionally very, very small structures in the nanometer scale. For example, this is the kind of stack of images we've obtained with the VSAM. It seems like it's moving, but it's not moving. You have here this structure, and you can follow it through the stack of images one after the other, and recognizing here the structures with the same quality of the transmission electron microscopy. So which is the best technique to imaging the brain? Well, the answer is it depends. It depends on three items you have here marked. These three items are completely related to one another. So you, first of all, have to think which is your goal. I mean, what do you want to visualize? Do you want to visualize one neuron, many neurons, or the dendrites, or the spines, or the synapse? This is the first item. But then, when you have decided what do you want to study and how you come to the resolution, if you are going to study a very, very small structure like synapse, you need the highest resolution, okay? But if you want to study neurons, you don't need so high resolution. Why? Because you are going to have lots of images, and you won't be able to manage them. So this is the second. And this is absolutely related to the scale. If you are studying electron microscopy, you cannot afford scanning the whole brain. You can't, I mean, because it's very time consuming. You won't analyze all the features in this. The same way, if you are studying many neurons, you are not using a small excuse me. You won't use electron microscopy to study neurons, but if you are studying synapses, you are not going to optic microscopy, okay, because you won't see them. So these three items are fundamental in the design of the experiments and what you are going to image. First of all, and depending on the goal you want to achieve, you have to use staining methods to visualize your neurons. So in optical microscopy, you have histochemical methods like the Golgi. You have immunohistochemical methods that allows you multiple labeling. Here you have an example like the one in the video, but here the neurons are in red, the terminals are in green. You can make colocalization studies, you can study diverse structures, and what we use in the lab to study the morphology of neurons are intracellular injections. There are many other markers like genetic labeling, tracers, fractional paths, etc. So intracellular injections is our material to obtain the images and then reconstruct neurons or dendritic spines. This is the setup where we perform the injections. This is a microscope that is linked to a micromanipulator where we put an electrode filled with a fluorescent dye. First, we have to stain the tissue to visualize the nuclei with DAPI. When you select your neuron of interest, we approach the electrode to the nucleus of interest, and then with an electric pulse, you make the dye, the lucifer yellow dye, go inside the cell. With this, you get your whole neuron filled with the dye, and you can visualize all the structures of the neuron, including the axon. And it also fills the dendritic spines, so this makes the technique very, very useful to reconstruct whole neurons, including spines, and the morphology of the spines, as you can see here. These injections of lucifer yellow can be made in many, many places on a single slide of your tissue, and you can compare, for example, between layers and study the morphology of the neurons present on each layer. As you see here in layer 2, neurons are quite different, I was going to say, but for you maybe it's not so different, but this kind of difference is with what we are studying, if these differences are relevant or not. Besides, it allows you to perform injections in coronal sections. If you have a block of tissue and this is layer 1 and this is layer 6, you can make injections and have this kind of images, or you can have horizontal sections through just one layer and perform the injections. You will have different reconstructions. In the first one, you have the whole neuron with the apical dendrite and the basal dendrites, but you will miss some of the branches that come out of the tissue. In horizontal sections, you will have the whole basal arbor, but you will miss the apical dendrite. So depending on the goal of your study, you will use one or the other. For example, this is a case where we use the intracellular injections to study different areas in the brain, on the cortical areas. So we perform injections in 3 areas: occipital area, temporal area, frontal area. And after injecting the region with several cells, we can study the morphology of the basal arbors of each neuron in each area, and we can study the spines on a single dendrite. So what's next? The next step is reconstructing the cerebral cortex. Why reconstructing? Because in my experience, most computer science engineers don't work with this kind of images. I mean, it would be nice, it would be perfect if I gave you this image, 3D image, a stack of images, and you say, well, you have here 1,033 spines, and the angles of the dendrites are 30 degrees, and so on. But that is not possible in my experience. So we have to perform a pre-analysis, that is the reconstruction of the neurons. Let's take the same case of the injections in the 3 different areas of the cortex. You have here the same slide with the field of the injected neurons. As you see, not all the neurons of the area are injected. I mean, the advantage of this technique is that you can select which neuron do you inject. So you have it quite separated from one another, and you can study the morphology very precisely. The disadvantage of, for example, immunocytochemistry is that you will have the whole area stained, so you cannot distinguish between one neuron and the other, and you won't know if one branch comes from this neuron or from this other neuron. So you can study, as I said, the basal airborne, or you can study the spines of the dendrites. So why I'm so insistent on the spines? Well, because the spines are, if you remember, the place where the synapses are made, so the number of spines reflects the number of excitatory inputs that the cell has. So if you count the number of spines, you will have an estimate of the number of synapses that that single cell or that single dendrite has. And this is very important when you are studying the transmission of information. How do we do the reconstructions? We have a method. This is an image of a maximum intensity projection of a neuron taking confocal. So this is the sum of the whole stack of images. And we have software that allows a perfect reconstruction of the neuron in 3D. This tool is called Neuroscience. It's a commercial software that allows you very easily, it's a very friendly program that allows you the tracing of all the dendrites of the neuron, the soma, the apical dendrite, and even you can detect and select the spines. So at the end, you have the whole neuron reconstructed in 3D. This program, as I said, is very friendly. You have two screens, one to move the neuron in 3D, the other goes plane by plane, and combining both screens, you will have your reconstruction. Besides, it makes a very nice analysis of the reconstruction, and you can take a lot of morphological features. Some of them are general values like the dendritic field area, because it's very important to know the area covered by the dendrites of a neuron, or you got the dendritic length, the sum of the length of all the dendrites, or you can study single dendrites and study the number of spines and the density along the, the length of the dendrite. And then you have another very useful tool that is shoal analysis. I don't know if you have heard of it, if you have worked with it, but basically it creates a number of nested concentric spheres centered in the soma, so you can study the morphological features in these spheres, whose radius you can establish depending on what you want to study. And here you have density of neurons on each sphere, of spines in each sphere. You have the total number of spines distribution. You have the complexity by counting the number of intersections with each of these spheres. And all these morphological features serve us to describe the neuron. In this study, we observed that the area covered by the basal dendrites of the neurons in each field is very different, as well as the total number of spines being lower in the occipital lobe and maximum in the frontal lobe. So these differences may indicate that the processing of the information in each area is very different. Also, you can study the differences between layers, as I explained before. So if you compare layer 2 and layer 5, we can see that the area covered by the dendrites is very different, and this will talk about the different integration of the information, as you must remember that the dendrites are the main input for the neuron. This is the areas. So this is the analysis we perform at the lab, but this analysis is useful for the other side of the other side of science, that is computing, informatics, and all this stuff that I, as a biologist, I have started to distinguish in this in this meeting. So I wanted to thank the organization for inviting me to talk, because now I have a little more idea of what is informatics, what is computing, and simulation, all that things. So what we have done is, with these reconstructions of the Neurolucida, we in the lab, we don't know, but the other people has told us that the information is contained in a series of nodes that are placed along the tracing, and this information is as big as you want. Apparently, you have information about the thickness of the dendrite, the position, the curvature, the angles forming, and so in this kind of skeleton, you have stored information, and then you can manipulate it. For example, we can create a soma. We trace the soma only the contour of the soma, but they have developed a program based on this contour and the thickness of the dendrite coming out of the soma. You can recover the real shape of the soma, and gives you a mesh, a triangular mesh, and you can study its volume, its position, and how the dendrites come out. This is useful not only for the analysis, but also for modeling a realistic shape for the soma of neurons. The same can be done with the dendrites, because you have here in the nodes stored information, all the information you need to recover a mesh representing the neuron. This has also been published as Neurons, and there are programs based in this first tool that are also very useful to recover these structures. So as we have seen, you can get a lot of morphological parameters from this kind of reconstruction, skeleton-based. You have the complete neuron or the basal arbor, and you can get the spine density. But what if you want to study in more detail the spines? As we say, the spines are the places where most of synapses are made. So it would be very useful to obtain the areas and volumes of spines, because the size of the spine is relevant for the strength of the connection with the axon input. So it can be done. You can obtain these volumes by using another different commercial program that it's called Imaris. Maybe you have heard about it. I hate it, but I use it every day. So what this program does is to perform shape. You can obtain a surface of each spine very, very precisely. This program allows you to obtain the length of the spines, the volume of the spine, and the position of the spine. Well, it's true that the length you have to calculate it manually. I mean, manually, you have to place a point on the head of the spine and another point in the neck. And you say, and you do it spine by spine? Yes, I do it spine by spine. 8,000 spines in a neuron. Okay, so yes, but when we have asked some informatics, please, could you develop a program that does? No, we can't, because human brain is very, very good at detecting differences. And our programs are very, very good on regular things, so no, it's better you do it. Thank you. So, we have this program that allows you to calculate volumes and shapes and areas of the spine. It has different modules. One of them calculates measures based on threshold intensity, and it's semi-automatic. You can establish the threshold you want to use, and it creates the surface around this intensity threshold pixels, and it reflects perfectly the shape of the spine. The problem is, as we have these confocal images, you don't have the same intensity in all the spines, so you have to create different surfaces to obtain all the morphologies of the different spines. But the parameters that this tool gives are very, very useful, and they can be used by informatics. I will say informatic people, so you understand that it's not only informatics, it's computing science and simulations and all the things, okay? And there is another module that is called Filament Tracer, that it's more automatic in the sense that it recognizes, you give a point of start and a point of end, and it recognizes the whole way of the dendrite, and if you, it's also able to detect the spines more or less. You can reshape them and edit the forms. It gives you a mesh based on concentrical circles, circumferences, and the center of the circumference is the axis of the dendrite or the spines. You have the positions of each spine, you have the volume, you have the length, you have a lot of parameters, but you don't recover the precise form of the spine. If you see here, this and this is the same spine. This is the mesh you obtain with the surfaces. This is the mesh you obtain with the filament tracer. It's not exactly the same. So depending if you want to study the morphology of the spine or just its volume, you can use one or the other model. What we have done with this IMARIS commercial program, we have studied the dendrites in different cases in human pyramidal cells, both in adult brain and aged brain, comparing the apical and the basal dendrites. So we studied over 8,000 spines, were 3D reconstructed in layer 3 dendrites of the pyramidal neuron. And this is how we perform the study. We take a whole dendrite from the soma to the end of the dendrite, capturing with the confocal microscope all the images contain the spines. This is a higher magnification view of a part of this dendrite, where you can see there are many, many different shapes of the spine. In this study, we were interested not only in the morphological values of the spines, but also in the shapes. So we can calculate the dendritic diameter, we can calculate the spine length manually, but we can calculate the spine density by counting the number of spines you have, and we can calculate the volume. The volumes, as I said, were captured for every spine, creating different surfaces and picking the one that better match the shape of the spine. This is an example of how the different intensity-based surfaces are created, and in this case we will select this one. We can cut this part of the shape and we have the spine. If you are interested how it functions or how it creates the surface, we can discuss it later because it's quite boring. And the results we obtain are represented here. This is the basal dendrites and the apical dendrites with the spines, color based on the volumes. The results were that apical and basal dendrites are very different, both in number of spine density, dendritic diameter, spine volume, and spine length. But what does this mean? This means that you cannot place the same number of spine in an apical dendrite or in a basal dendrite. So it's important to know what are you putting in your model so that the neuron functions. And it's not the same if the specimen is adult or it's in this case, I think it was a male, 80 years old male compared to a 40 years old male. So there are differences, and these differences in spines may reflect differences in function. Why? Because as I said, the neurons, the size of the neurons is related to the current that goes through it. So at the end, what you want to have is a map of the different currents that you have in your spines, and this may be useful to know the function of your neuron and your simulations. And that's why we are not dumb persons yet painting spines just for fun. No, we think that this is important to have precise data with which you can perform your simulations. With the other module, the filament tracer module, as I said, allows you to have the precise position of each spine. So we thought it was a good idea to have the whole reconstruction of a cell and compare the features within one cell. So you can have more information about how to place spines, where to put them, and so on. This was made. In fact, I did this neuron. I know this neuron by its shape. I think I recognize every spine. And with these meshes, if you remember, there were circumferences. You can obtain a program that allows to explore these differences between the spines in a single neuron. You have a lot of features you can compare, and it has been published as Pyramidal Explorer. So the idea is to part from an image and through mathematical calculations have an in silico reconstruction of your neuron that you can manipulate or study. I think we are in the right way because many articles have been published using these meshes to obtain features about the distribution of the spines in the neuron, comparing each branch, so you have easier So why is this important? This is important because what we want to know at the end is the connections of the spine. And I think I've been too slow, so I'm going faster to talk to you about the electron microscopy and the VSAM, and why it's important the synapse, because this is the way you have the transmission of information. You want to study synapse, you have to go to electron microscopy because you have this relation between goal, scale, and resolution. With traditional electron microscopy, you are very slow in your work. So we use the VSAM that, as I said, have two beams, one that mills the specimen, the other captures the images, and at the end you have holes in your tissue and a stack and the image in the wall at the end. And you have your stack of images that using a specific software you can use to reconstruct bricks of small portions of the brain and study their synapses or the element you want and study the shape, the volume, the relationship between the synapse, the size, etc. This has also given place to some publications using this software that this is not commercial, has been developed specifically to our lab. It's called Spina, and I think some of the students are going to talk about it. And at the end, I think we are in the right way to get to a model of neuronal cells. Thank you very much. This is all the people in our lab. We are a lot of people doing similar things, not all the same. And if you have any question, easy, please.